Hello student, today we start the BSc second year, that is paper 7, Fish Developmental Biology. Okay, BSc second year, paper 7, that is Fish Developmental Biology. Within that, unit number 1, Developmental Biology. Within that, the types of eggs, gametogenesis, types of eggs, fertilization of egg, and cleavage already we are completed. Okay, then we started last class, that is unit number 2, that is developmental biology. Within that, number 1, that is morula, already we are completed. The next one, that is blastula. Okay, unit number 2, beat number 2, that is blastula. Before that we start the blastula, from beginning we will see about the male female gamete, the forms zygote, then removal of yolk. All the process means division of cells or increase in cells. Then the about the cleavage, how can they form the cleavage, that is the division of zygote in small unit. Then the start usually within the 30 minutes. Total number of cleavage, that is the 6. Then cleavage become blastodermic cap. All the process in the sense, shortly it says the cells increases. Then about the cleavage, fertilization of blood tumors, then the mitotically 2 cells, then the cleavage fertilizing to 4 cells, then the 8 cells. Within 3 hours, within 3 days, fertilization take place, and that is the 16 stages. The all the process in the form, that is form of morula. The morula, then next stage, that is the blastula. Today we will see about the blastula. This is the process. Okay, means development of stages of fish. About the today we'll see about the blastula. At the end of morula, already I told that at the end of morula, a space is called the blastocele. Where is blastocele? Here. It's the blastocele. At the end of morula, a space called blastocele is a form between the blastoderm and periblast. Where it's formed? Here. Periblast and blastocele. Okay. At the end of morula, a space called blastocele is formed between the blastoderm and periblast. Okay. The embryo in this stage represent a blastula stage. This is the blastula stage. The formation of blastocele. Here the blastocele. This is the periblast and this is the blastocele. The formation of blastocele is followed by enlargement and flattening of the blastoderm. Okay, so that the radially symmetrical embryo assume a bilateral symmetry. Is bilaterally symmetrical. The next point, the blastoderm gives rise to embryo. Important thing, the blastoderm gives rise to embryo, while the cell of the periblast, this one, probably serve to digest the yolk. Probably periblast serve as a digestive yolk and supply it to the developing embryo. This one. The central thicker part of blastoderm consists of cells destined to form the embryonical material. This one, the embryonic material represents. Its distal portion marks the prospective posterior end. Here. And the central part corresponds to the further anterior end of the embryo, anterior and the posterior side of the embryo. Dividation of cell, increase in the cell, all the process is going on, that is in the blastula. Okay,